I gravitate, just 99% whole foods, but I feel that that is largely not a problem, and is an indicator. High omega-6 content in your blood and in your dietary pattern is indicative of the type of diet you are eating. So generally speaking, I would not, buy seed oils and use them for cooking and things like that. Like soybean oil, obviously canola oil. We do have avocado oil, and we have, extra virgin olive oil. I don't heat that. And we do have, I think they're the only two oils that we have, and we largely cook with butter and lard. But I don't think it's a problem if your diet is good. And I think where we come into the problem is where if we add these oils to heating the oils is a major problem. And then an overconsumption of these oils, like in a surplus diet. And what will indicate whether they're a problem or not is when you simply do like an omega quant test. And I've seen some pretty scary omega quant tests with people with super high omega-6s that didn't know.
So this is a test you can buy that measures your omega-6 to omega-3 levels.
Yep. And I actually believe, I've probably given talks over the last 2 or 3 years, and I advocate for this to become part of the standard of care for blood work. Like when you get a comprehensive metabolic panel, that your blood levels of omega-3s and omega-6s should absolutely be the epidemiological, not only the epidemiological data, but the data in general on blood levels of these fats and the ratios of these fats are like a better indicator of overall metabolic health and your risk than many other things that we're looking at.
And most people will not have gotten these numbers. So, whether you're using like the at-home blood testing stuff, or you're going to get your labs done, or just a doctor's checkup where you're getting, your blood drawn and your quote blood work done, usually those tests will not look at omega-6 and omega-3s.
Yeah, it's really rare. And, I got turned on to this by maybe 10 years ago by Rhonda Patrick, and she's hosted a number of guests on there that talked about it. I was convinced this is important. My early postdoctoral work was basically really focusing on PUFAs, and I could tell you in the lab, the amount of PUFA, when you eat PUFAs, it just goes right to the plasma membrane, the lipid bilayer. And the higher the concentration, the PUFA concentration in the plasma membrane of your cells and in your neurons in particular, the higher potential for oxidation. And then when we talk about inflammation, we are basically talking about oxidized lipids. So we do a T-bars test and measure something called malindialdehyde. But docosahexaenoic acid, that has like 6, I think, double bonds. Like, these are susceptible to oxidation.
Right. So the polyunsaturated fatty acids, they're named after the fact that they have multiple double bonds.
Yeah.
And it is those double bonds on the fatty acids that can be oxidized, and our body's filled with oxygen, obviously. Mm-hmm. And that basically, when that happens, when those PUFAs are oxidized, it produces toxins. And we tend to basically just call that inflammation. Yeah.
And that is dangerous. Like I taught, redox biochemistry and biochemistry, and I'd go do the whole pathway, and it just becomes, when you start to go down the rabbit hole of the biochemistry of membrane lipid peroxidation, it's like a futile cycle, and it becomes scary if you do not have the endogenous antioxidant capacity to manage that.
Yeah.